In this video lecture, we're going to begin to discuss glucose catabolism in a little bit more detail. So as I've mentioned in earlier video clips, the story of the catabolism of nutrients inside a chemoheterotrophic type organism generally starts with glucose. And here's what glucose looks like. It's a sugar molecule, a monosaccharide. It has 6 carbons in it. There's 1, 2, 3, 4, 5, and 6. It also contains 6 oxygens and 12 hydrogen atoms, and they are arranged in this particular pattern. So this is the preferred energy source for most chemoheterotrophic type organisms. And then we've seen this before as well. Okay, so we've taken a look at this before. This is the overall. We're going to be discussing a process called cellular respiration here in Chapter 7, which you hear about in every biology class. And this story starts with glucose. There's a glucose molecule. Now, we've talked about this before, the inputs and the outputs of cellular respiration. So we've got our inputs over here, and over here we have the outputs. Now, we've got an arrow there, and just keep in mind there are many steps that use many different kinds of enzymes involved in going from these inputs to these outputs over here. But when you finish this process, glucose, right in here, is broken apart. It is catabolized. It gets oxidized as it's being broken down. And the carbons, you've got 6 carbons in every glucose molecule that goes through this process, right? Where do those carbons go? So they can't just vanish. They've got to go somewhere. And as glucose gets broken down, those carbons all end up in CO2 molecules. So they're going to get converted to carbon dioxide. Remember how we talked about during oxidation reactions, extra oxygens will bond to so as these carbons are being oxidized as part of that process, more oxygens attach, and that's how you wind up with more oxygens here than you had over here as well. You also have 6 oxygen gas, O2 molecules, that are inputs for this process, which are required if you do aerobic cellular respiration. Remember, aerobic organisms require oxygen gas for survival. And then now we've been talking about how ADP and phosphates get bonded together to build ATP. So you need those as inputs. And over here you see the ATPs as outputs. So as this molecule, this glucose, gets broken apart and converted to carbon dioxide, that's releasing energy that is driving the linkage, the bonding of these ADPs and phosphates together. And then we'll also see water is an output of aerobic cellular respiration, good old H2O, and heat is an output as well. And the reason for that, again, is as glucose gets oxidized, broken apart, not all of that energy is harnessed to form ATP. Instead, some of it winds up getting lost in the form of heat. There's another process that we'll talk about, and you may have talked about this in your A&P1 course when you discuss the muscular system, because your muscle cells can do this process called fermentation at times as well. Fermentation is an alternative process where glucose gets partially broken down or oxidized. It gets converted into 2 molecules called pyruvate that have this chemical formula, C3H3O3. Then you still have ADP and phosphates as inputs with some ATPs over here as outputs, and heat is also given off as well. And then as we'll see, these guys wind up getting converted over to waste molecules that are generally acids or alcohols or gases in some cases. So this is an alternative process. Now up here, what's the number of ATPs that a cell generates by doing cellular respiration starting with 1 glucose molecule? It's going to vary from organism to organism, but we'll take a look at some of those numbers. Fermentation, a cell generally gains just 2 ATP molecules for every glucose molecule that's broken down. So it's not going to be as efficient as we'll see. Okay, So a little bit more about oxidation and reduction, because you guys are going to be learning about this as you do your reading in Chapter 7, or this terminology is going to be used quite a bit. And again, I don't want you guys stressing out too much over a lot of the chemical terminology. Follow along with what we're covering here in the video lectures and use your study guide and learning objectives for Chapters 6 and 7 as a guide. But as glucose is broken down, and other types of organic nutrients as well, a lot of the chemical reactions that take place are called oxidation reduction reactions. And so we've already talked about when a molecule like glucose gets oxidized, it is losing electrons. That's the definition of oxidation. And those electrons are actually contained within hydrogen atoms. So what they're showing you over here, you've got an organic molecule that could be glucose. It could be one of the breakdown products of glucose. It's being gradually broken down. And when that molecule becomes oxidized, it's losing a couple of electrons that are shown there in hot pink. But those electrons are part of hydrogen atoms. So really what's happening is two hydrogen atoms are being plucked off of that molecule. Those electrons have to go somewhere, though, so they don't just vanish. Electrons don't vaporize. They've got to go somewhere. If they go off of one type of molecule or atom, they've got to go to another one that's going to pick them up. And so when these oxidation reduction reactions take place, anytime a molecule is oxidized, some other molecule is going to become reduced. And reduced means when a molecule becomes reduced, it acquires electrons that it did not have before. So reduced, if you think about if a molecule picks up electrons have a negative charge, so the overall charge of that molecule is now lower. It's going down, it becomes more negative, and that's where the reduced terminology comes from. So as you guys will see, as you go through these processes that are discussed for catabolism, there are electron carrier molecules that are involved in these processes. And the most common ones are NAD+, and that's an abbreviation for nicotinamide adenine dinucleotide, sorry. What did I do with here we go, looking for my stylus. I apologize for the quality of the writing on some of this. I'm writing on an iPad, and they have not yet invented a stylus that can write legibly on an iPad for some unknown reason. But anyway, this NAD is an organic molecule, and it's actually a coenzyme. So very often enzymes have little accessory molecules called coenzymes that are required for them to be able to function properly. And so as these hydrogens and their associated electrons are plucked off of this molecule, they attach to the coenzyme over here. So this molecule was oxidized, and this one was reduced. So now here is your oxidized organic molecule. Your NAD+ becomes something called NADH because it has acquired a hydrogen atom. Erase all that. It acquired one of the hydrogen atoms off of the original molecule and its associated electron. You can see that right there. And then the other hydrogen atom was also plucked off, and the electron of that hydrogen atom wound up on the NAD. The proton, so a hydrogen atom, if you guys remember from chemistry, is one proton and one electron. So if the electron from that other hydrogen atom has bonded to NAD, you basically just have that proton left over, and that proton is also essentially a hydrogen ion, a positively charged hydrogen ion that we write like that. And that one remains unattached to the NAD. So the NAD+ over here, it had a positive charge, and it has acquired a neutral hydrogen atom and also an extra electron. So it no longer has that single positive charge. It is now a neutral molecule that we call NADH. Okay. So that's oxidation and reduction types of reactions that are very common as glucose and other types of nutrients are being broken down. All right, so this is a figure from a previous textbook, which I like a little bit better than the one that's in your textbook, and so I have left this older figure here in my video lecture. What this is showing you, it's showing you an overview of the different processes that organisms use for glucose catabolism. And over here on the left-hand side, we have respiration, and that is not breathing type of respiration. This is cellular respiration, which is the complete oxidation of glucose. You completely break it down, harness the energy to make ATP. And then over here on this side, we have fermentation. Fermentation is going to be an alternative type of process. And so up here at the beginning, here's our glucose that we're starting with. And notice that we have this process right here called glycolysis. Now you've heard about glycolysis if you've taken a biology class before. Glycolysis is the partial breakdown of glucose. It gets broken apart into two molecules called pyruvic acid. And so glucose is a molecule, sometimes this is written just thinking about the number of carbons that are present. And during glycolysis, glucose, which has 6 carbons in it, gets split apart into two molecules of pyruvic acid, each of which have 3 carbons. Though they also have oxygens and hydrogens, but usually we just think about and talk about the carbon backbone that's involved here. This process is called glycolysis. Glyco refers to glucose. Lysis means to split something. So you're splitting glucose. Erase all of that. All right, another thing I want to point out over here, and we're going to talk about glucose in a little bit more detail shortly. I'm sorry, glycolysis in a little bit more detail shortly. During glycolysis, notice we've got an ATP over here as an output, so we'll talk a little bit more about that. And notice on the diagram you've got some NADHs over here as outputs as well. And again, the NADHs got a hydrogen and an electron that were originally on our glucose. Our glucose is being oxidized. So a hydrogen atom and an additional electron that were originally on the glucose are transferred over to NAD+, and NAD+ becomes NADH. We'll talk a little bit more about those as we go through the story as well. All right, so once you have these pyruvic acids at the end of glycolysis, now you have an option. You can go one way or the other. One way that you can go is over here, and you can do fermentation. That's one process that can be performed, and we're going to talk in quite a bit more detail about fermentation. It's a very important process for microbiology. The other option, though, is that you go through these processes over here and complete cellular respiration. So pyruvic acid will feed into a process that's called the Krebs cycle, and that's also known, if I can write, hopefully I can write on here and it not look like I'm a kindergartner. The Krebs cycle is also called the citric acid cycle. And then after that, there's another series of chemical reactions, another process called the electron transport chain that takes place. And those two processes together lead to a lot more ATP being produced. Because remember, our goal here is to break apart this glucose and make ATP. We're going to harness the energy out of the glucose and store it in these ATP molecules, because those ATP molecules are going to function like the gasoline for the cells. But we have two alternative processes that can be used. Both of them have glycolysis in common, and then you've got an option to do respiration or an option to do fermentation. Okay, so a little bit more about glycolysis. So again, glycolysis is the first phase of both cellular respiration and fermentation. So it's a pretty important process. And now we just talked about how during glycolysis, a single glucose molecule is split into two pyruvic acids. Each of those has 3 carbons in it. And is that a catabolic process? Hopefully you're thinking yes. And what do we know about catabolic processes? Those release energy. Some of that energy is harnessed, and it's used to take ADPs and phosphates and link them together to make ATP. So that's the whole goal here. We're trying to make these guys. We need to make as many ATPs as we possibly can. So that sounds pretty straightforward, but it's actually a process that has 10 different steps, as you can see on this diagram over here. Those different steps will each use a different enzyme. So you've got lots of different enzymes that are involved in this process as well. This is a diagram showing you a little bit more detail. There's glucose up here at the top, and these are what those 2 pyruvic acids look like down here at the end. Another thing I want to point out on here is notice on this diagram right there, they are showing you that 2 ATPs are being input into the process. And what that means is you actually burn through, for every glucose molecule that enters glycolysis, 2 ATPs will be broken apart. So those ATPs will be converted to ADP and phosphate. That's releasing energy that is used to get this process started, get glycolysis underway. So a lot of times that's referred to as an energy investment step. Sometimes people make the analogy, it's like when you strike a match to start a fire, you've got to use some energy to strike that match, to rub it against the concrete or whatever, to make it light. So it's kind of like lighting a matchstick. It takes some energy investment to get the process started, and then the process will be able to proceed to completion. Now, as you look down here, for every glucose molecule entering the process, there are 2 NADHs as outputs. And again, those NADHs started out as NAD pluses. They are acquiring a hydrogen atom plus another electron originally contained up here in the glucose. So they're temporarily, as we're going to see, they're temporarily storing the hydrogen and the extra electron for a later purpose during cellular respiration, as we'll see. But again, that's part of catabolism. You have plucked these hydrogens off of the glucose and stuck them onto NAD plus molecules temporarily. And then notice also, let me kind of clean this up a little bit. Notice also we have 1, 2, 3, 4 ATP molecules that are coming out as outputs. So you've partially broken apart this glucose. You've been plucking hydrogen atoms off of it and breaking this molecule apart. And energy was released by those processes. That energy has been harnessed to form 4 new ATP molecules for every glucose that's broken down. However, remember we used up 2 ATPs to get this process started. So what's the overall net gain for the cell? You use up 2, you gain 4. The cell winds up gaining 2 more ATPs that it did not have before it started doing glycolysis on that single glucose molecule. So that's a key output of glycolysis, 2 new ATPs for the cell for every glucose molecule that enters and completes that process. All right, and then as I mentioned earlier, as glucose gets broken into those 2 pyruvic acids during glycolysis, that's glycolysis. Multi-step process, 10 steps, 2 new ATPs for the cell. Those pyruvic acids will then feed into either cellular respiration or fermentation. And as we'll see, not all microorganisms can do fermentation. Some can and some can't. That'll actually be the basis for some of the lab tests you run on your unknown bacterial organism during the lab simulations. Then also, actually, some organisms can't do cellular respiration, as we'll see. Some organisms can do both. So it's not always truly a choice. Cellular respiration involves the citric acid or Krebs cycle and the electron transport chain, and generally produces a lot of ATP. Fermentation actually does not produce any additional ATP. So for every glucose that enters the fermentation step, you're only getting 2 new ATPs for every glucose that you start with. And instead, what happens is fermentation leads to the production of acids and alcohols or gases as waste products, and those come from the pyruvates or pyruvic acids. Pyruvate and pyruvic acid are interchangeable terms for our purposes. These guys get converted into acids or alcohols and or gaseous wastes as part of the fermentation process, and we'll talk quite a bit about more of that when we have a separate lecture on fermentation. All right, so that's just kind of an introduction to glucose catabolism. In the next lecture, we'll home in more on what's going on with the cellular respiration option that can occur after you do glycolysis.